Right. So basically, I was asked by Lauren to give you a short summary of our recent paper published in Blood on the role of secreted mutant calreticulins that appear to work as rogue cytokines in myeloproliferative neoplasms. So this is an international study with samples from Japan, from Italy, from Austria, from France, and from Belgium. And you may remember that several groups, including ours, have published in 2016 and later on the fact that these mutant calreticulins, discovered by Robert Kralovich and Tony Green in 2013, induce essential thrombocythemia and myelofibrosis dependent on expression of MPL, the thrombopoietin receptor. And so what happens is that the mutant calreticulin becomes very specific to this thrombopoietin receptor extracellular domain. And then they meet in the ER, they are transported through the Golgi to the surface. And I think Stefan also has done a lot of work on that. And then they are exposed at the surface, and we shown in 2019 that, in fact, you need the complex on the surface. If it's not on the surface, it's not oncogenic. And that's good news for therapy. Because then it's accessible. So the questions that we asked in this study were whether or not these mutant calreticulins are also secreted, not only exposed on the surface, especially from cells that don't have TPO receptor. If they are secreted, are they secreted alone or in a complex? Are they stable in plasma? And whether they are in plasma, do they signal via TPOR on other cells, on all cells that have TPOR, or just on cells that are from the clone? And so we, in this international consortium, we got samples from 111 MPN patients with calreticulin mutants, 35 with JAK2 mutation, triple negative 2, and healthy control 11. And via an ELISA, worked out essentially in Robert Kralovich's group and at the Myelopro in Vienna, we were able to test this mutant calreticulin. You see that you detect it, and that we can have patients with 100 or 60 or 80 nanograms per ml. The average is around 26 nanograms per ml. There is not much difference between type 1 and type 2, but the other more rare ones are less secreted. There is a very nice correlation with the allele burden. And there is more protein in myelofibrosis than in ET, and we cannot really make a statement between myelofibrosis and pre-myelofibrosis patients. But clearly the protein is detectable. Now, we wanted to know the source of cells, and for that we turned to our mouse model. So we created a knock-in mouse model in the lab that is either heterozygous del52 on the normal background B6 mice, or on MPL minus mice. And you see here in the supernatant, we can IP and detect mutant CALRs similarly from del52 knock-in, whether or not they have MPL. So clearly it's secreted in the absence of expression of MPL. And if you look in the cells, in the blood cells, you can certainly see that if you don't have MPL, you release much more. So it's secreted essentially from clonal cells that don't have MPL. And we're working out now whether they're the monocytes, the granulocytes, the B cell, we don't know exactly. Maybe all of them, or maybe one of them is very strong. Now, what we have looked then is on the right here, we have the half-life in plasma in patients, and that's 426 minutes. So it's rather stable. However, if we take recombinant mutant CALR and we dissolve it in medium or in healthy plasma, half-life is around 30 minutes, between 30 and 35 minutes, depending on which medium you have. So clearly, it's much more stable when it's secreted. And the question is how is it stabilized? And for that, we looked by immunoprecipitation and mass spectrometry, and we identified in 5 patients that the most important protein in the complex with which it's secreted is the extracellular domain of transferrin receptor 1. And that was a surprise, but it's a very robust result. That is shed somehow. And if you look here on the bottom, you see that in UT7 cells, you have mainly the full-length transferrin receptor 1. But if we express the DEL52, then half of it is cleaved. So the presence of DEL52 stimulates shedding of the extracellular domain. And if we mix the extracellular domain of transferrin receptor 1 with the CALR DEL52, we can get from, we can increase the half-life, but not to the 400 minutes. So there must be also something else. It's not enough. And then we looked at what is the percentage of the mutant CALR in the plasma bound to the transferrin receptor, and it's really substantial. It's between 1/3 and 99%. So it's a lot. And interestingly, it's known that one of the asparagens of the transferrin receptor remains immature even outside the cell, and that's via what this binds. Nevertheless, this complex, as I will show you later, can still bind to TPO receptor. So it's not blocking the binding to the TPO receptor. Now, we then looked whether this recombinant calreticulin mutant can stimulate signaling. And to make a long story short, this was a very paradoxical result for us that we didn't know how to interpret. It took us years to figure it out. So in fact, what happens is that you can see signaling only on cells that already express mutant CALR. So if you just take cells that have TPOR, the recombinant mutant CALR starts signaling at 100 microgram per ml, which is huge. But at low level, which are compatible to the 50 nanogram per ml, 100 nanogram in the plasma, only the mutant carrying cells respond. So TPOR is not sufficient. You need to have endogenous mutant CALR together with a receptor on the surface, and that allows the recombinant to oligomerize with the mutant CALR and activate the immature receptor that is on the surface. So basically, it stimulates only the clone. And if you look here, we have the EC50. It's 99 nanogram per ml. Of course, it's higher than the TPO itself, but the level of signaling, in the mutant, you see here in panel D, is higher than even with TPO. So this is a substantial signaling stimulation that you get on the clone, not on cells that only express TPOR. And that you can see phosphostat 5 activating in cells that are CRISPRed for the mutant CALR, and also in the knock-in mouse bone marrow, but not in cells that only express TPOR. So the question is, of course, why? And I will just focus on the bottom part, especially on panel D. So here we took cells that have CRISPRed murine CALR DEL52, which migrates at a lower molecular weight. And on these cells, so either those are CRISPRed on the right or wild-type CALR on the left, and then we add an exogenous human CALR DEL52, which migrates a bit heavier, and then we take these cells, we wash them out, and then we reveal the bound human recombinant CALR DEL52. And you see that if you already have endogenous mutant CALR, you have much more human CALR binding to it. So this is the result of the oligomerization ability that the group of Komatsu and Akira showed in the beginning, that this mutant CALRs oligomerize strongly with each other. And the only cells that will have immature TPOR are the cells of the clone, of course. And finally, we took bone marrow cells from patients, either with CALR or JAK2 mutation or healthy controls. And then we checked whether the recombinant human CALR could induce TPO-independent megakaryocyte colony formation. Now here we looked at half-life in the medium for these colonies, and in 35 minutes. So we had a mathematician calculate for us how much do we have to add once to have an overall 2-3 days of 100 nanogram per ml, or how much do we need to add daily to maintain the level, because the half-life is very short. And you can see that if you add just once 20 microgram per ml, and then you have this overall average concentration, which is 50 nanogram per ml, then you see that you have stimulation in the patients that already have CALR mutation, not in the JAK2 V617F patients, not in the healthy controls. And you can also see that we detect it also when we add every day to reach these levels. So this can have a functional consequence. And what we concluded from this is that the mutant CALRs are secreted, especially from the cells belonging to the clone that don't normally express TPOR, because TPOR would retain it at the surface. They are secreted in complex with the extracellular shed domain of transferrin receptor 1, and that the mutant CALR stimulates this shedding, and that at the level similar to those in patient plasma, the mutant secreted CALR only stimulate the clone. And finally, these circulating levels will be important for all the protein therapeutics, because they can somehow trap the protein therapeutics or redirect it to other places. And I think it's a very interesting phenomenon to be studied further. So that's it.
Interesting, very interesting. Do you have any experience on this secreted CALR in patient after allogeneic bone marrow transplant?
No, because we don't even have any in our clinical hospital in Saint Luc with progression in CALR, and they're more rare, right? I mean, I don't know. Maybe Stefan, you have some, right?
Yeah, we can talk about that.
It would be a nice biomarker, right? I mean, you can check.
I think this is really interesting. We had, as you said, done some secretion studies and found that if we stimulate wild-type cells, we didn't see any stimulation there, but this explains it. So we didn't have the mutant cells. Can I ask you something about the mice that you have? Have you looked for circulating levels of mutant CALR in those? And
yes.
Shouldn't, because you're saying this may affect antibody treatments of in mice, shouldn't this have inhibited the activity of these antibodies in vivo?
Well, it depends, because if you have only 20 nanograms per ml, and most of the patients are around those, it could be that the pharmacologic levels of antibodies that people will inject, for example, Insight and other people will have antibodies. There are 3 or 4 companies doing it. Yeah, they're much higher. So, so it may be that they're rare patients with a lot, and you have to be careful there.
Mm-hmm.
But what we do not know is whether this high level, it just reflects a little burden, or it modifies disease somehow. We don't know. And we don't know whether this mutant CALR protein could go and bind to some other cells, right?
Right.
You may mean they would bind to some kidney cells or whatever. I mean, then it's not so good, right? So we don't know. But the fact that we have it with transferrin receptor 1, and it's not completely naked, being able to touch everything, is good news.
Mm-hmm.
Because if it would be sticky and bind to everything, that's going to be a problem, right?
Yes.
Stefan, TFR1 is a normal target of normal CALR in its chaperone role. Usually,
it has not been reported before.
Okay.
We don't know, because we were just looking now at some knockout CALR cells to see how this. It would be like calnexin. Usually, calnexin takes care of the transmembrane proteins that are glycosylated, and calreticulin of the soluble ones. So it may not be a professional client of calreticulin, but for sure is of calnexin.
Okay, thank you. Anyway, fascinating results, right?
Yeah.
So what I wanted to say is that we have now a paper coming out in Nature Communications where we explain finally why TPOR is the only receptor that binds to it. That is because of 3 patches of negative charges, 2 and another one, which are bound by the positive charges in the tail. So we found finally the chemical explanation, because there are thousands of proteins that are glycosylated, right? Including the transferrin receptor 1. But what TPOR has special is some negative patches, which we show now structurally to bind with the positive charges. And that's why the affinity is so much, and you have this dimerization that is permanent.